A warm welcome to one and all. Myself, Dr. Rashmi Joshi Sawalkar, Assistant Professor, Department of Zoology, Shri R.L.T College of Science, Akola. Dear friends, today we are going to learn the lesson cell membrane and the different models of cell membrane. So, this is the chapter from your semester 2, unit number 1, and before this we have studied the two chapters. This is the third chapter of our first unit of the paper Zoology, Cell Biology and Developmental Biology. So let's start and see the different models of cell membrane. So the outline of this lecture is: what is cell membrane? What is cell membrane? Chemical composition of cell membrane, structure of cell membrane, then different functions of cell membrane, then models of cell membrane. This is the main part of today's our lecture. So let's see one by one. What is cell membrane? The cell membrane, also called the plasma membrane or plasma lemma, is a biological membrane separating the interior of the cell from the outside environment. So it is a cell membrane which separates the interior of the cell from the outside environment. It appears in thin sections with the electron microscope as a triple layered structure and about 7.510 nanometers thick. So the thickness of plasma membrane under the electron microscope, it can be observed like a triple layered and 7.5 to 10 nanometer in thickness. This term coined by C. Nageli and C. Kramer in 1855, and the name plasma lemma is given by the scientist J. Q. Plough in 1931. Now, what is cell membrane? The cell membrane, which is flexible and allows a unicellular organisms to move. See, it is the unicellular organism. As they do not have any special locomotory organs, they locomote with the pseudopodia. So the outermost flexible cell membrane is flexible, which forms the pseudopodia for locomotion. Now, chemical composition of cell membrane. Let's see what is the chemical composition. So the first element which is a component of plasma membrane is lipids. Four major classes of lipids are commonly present in the plasma membrane. Number one is phospholipids, which is most abundant out of the four lipids. First is the phospholipids, second is sphingolipids, third is glycolipids, and sterols or cholesterol. All of them are amphipathic. Cholesterol is abundant in mammalian cell and is absent in prokaryotic cell. Point to be noted that cholesterol is abundant in mammalian cell and it is absent in prokaryotic cells. Now the second component which makes the cell membrane is the proteins. So plasma membrane contains about 50% of proteins. Total amount of elements are there from which protein percentage is 50%. Amount and type is variable. Myelin cells contains about 25% protein. Internal membranes of chloroplast and mitochondria contains 50% of protein. Proteins of plasma membranes are of two types: ectoproteins and endoproteins. Proteins which are present in the plasma membrane are of two types. The first one is ectoproteins and the second is endoproteins. Plasma membranes contains structural proteins, transport proteins, and enzymes. Some of them acts as receptors. I have explained you this property of these types and properties of protein of plasma membrane in the previous lecture. Now, enzymes present in the plasma membrane are acetyl phosphatase, acid phosphatase, ATPase, RNase, maltase, lactase, and phospholipase, etc. The third component which is present in the plasma membrane are carbohydrates. They are present only in the plasma membrane and are present exterior. that is glycoproteins or polar ends of phospholipids at the external surface of plasma membrane. These are present externally on the polar heads. They are attached, the carbohydrates are attached, and those are called as glycoproteins. Now let's see what are phospholipids. Phospholipid molecules has a polar head and non-polar tail. The polar head is charged and hydrophilic, water-loving, and it is charged. Facing outward contains the phosphate group and glycerol. The non-polar tails are hydrophobic, they hate water. Facing inward contains two fatty acid chains that are non-polar. So the head region is polar and hydrophilic, and the tail region is non-polar and it is hydrophobic. When phospholipid placed in water forms spherical bilayer. This is the structure of phospholipid. See here, this is the chemical composition structure given here. Double bond, cis double bond is there. We want only this structure. We want to study here only this structure or this structure. No chemical composition is required here. So let's see, this is the phospholipid structure. This is the head region, which is polar end, or it is and the tail region, which is non-polar end. It is charged and it is not charged. Tail region is not charged. So this type of phospholipid structure is present here. Now membrane cholesterol. The amount of cholesterol may vary with the type of membrane. The membrane is of which type, so it will decide that how much amount of cholesterol is present there. So the plasma membrane have nearly 1 cholesterol per phospholipid molecule. Per phospholipid molecule, 1 cholesterol molecule is present. So this is yellow colored cholesterol molecule, this is phospholipid. So see here, every phospholipid molecule showing presence of 1 cholesterol molecule. Now proteins are critical to membrane functions. So what are the functions of protein and which are critical for the membrane function? So see here, the green color structure, 4 different structures of proteins are shown here. The first one is structural protein, and it is bound to the one side of the plasma membrane. It is the structural protein. B is the recognition protein. See, lock and key model is here, and it is transmembrane. From inner layer to outer layer it is present, and that type of protein is recognition protein. This is the transmembrane protein, which helps in communication, and this is also gate channel protein, which helps in transportation. So structural support, recognition, then communication and transportation, these are the functions which are performed through the proteins present in the plasma membrane. Now structure of cell membrane. So structure of cell membrane, this is the fluid mosaic model, colorful diagram. These are the protein molecules all, and this is the purple color is the carbohydrate chain which is attached to this protein molecule and on the outer surface of the plasma membrane. Then this is the lipid bilayer. This is the inside of cell. This is the outermost environment of the cell. Now membrane composition components. So these are the phospholipids, 2 bilayer that I have explained you in the previous lecture how this bilayer is formed. This middle portion is water hating, and this polar ends, head region, are water loving. So this type of structure, sandwich structure is formed here, bilayer structure is formed here by the plasma membrane. So phospholipids are present, this yellow colored cholesterol are present in between the membrane, green color proteins are present, and on the proteins on the outermost side the glycocalyx chains are present, sugar chains or carbohydrate chains are present. Now inside the membrane, in the inner layer of plasma membrane, cytoskeleton is seen. These are the fibers, actin or myofibrin filaments. Then peripheral protein is here, integral protein is here. So this type of membrane components that we have seen in the previous two slides. So these are the membrane components and they combine to form the plasma lemma structure. Now functions of cell membrane. So functions of cell membrane, plasma membrane, or the outermost wall are: keeps a cell intact. Plasma membrane keeps the cell intact. It is protective barrier, that is protection. Then regulate transport in and out of cell. Selectively permeable means selected solute and solvents are transported from inner side to outer or outer to inner side. So that's why it's called as selectively permeable. Permeability is for selected particles, that's why it is called as selectively permeable. Small lipid soluble molecules, example oxygen and carbon dioxide, can pass easily through this membrane. Water can freely cross the membrane. Water can freely transport from one outer layer to inner side and from inner to outer side. Now ions and large molecules cannot cross without assistance. The large molecules and some ions, for example magnesium, sodium, potassium, that type of ions cannot be transferred without any assistance. Assistance is required for their transportation. Then allow cell recognition, provide anchoring sites for filaments of cytoskeleton, provide a binding site for enzymes. Interlocking surfaces bind cells together, that is junction. Contains the cytoplasm fluid in cell. These are the functions of plasma membrane. Now membrane structure. Let's see here. Membranes consist of a phospholipid bilayer combined with a variety of proteins in a fluid mosaic arrangement. This is the type of fluid mosaic model. So fluid mosaic arrangement, the surface of cell membrane are hydrophilic, water loving, the interior are hydrophobic. See, this is the phospholipid bilayer and hydrophilic region of protein, that means water loving region, and this middle portion, the tail part, shows it is the hydrophobic region of protein, hydrophobic region of plasma membrane. Now models of plasma membrane. Today's the important part we have to study here, that plasma membrane was studied by the scientist or discovered in many different models. So let's see one by one the models of plasma membrane. So the first one is Danielli and DAVSON model. So Daniel and DAVSON, in 1935 Danielli and DAVSON studied triglyceride lipid bilayer over a water surface. They found that they arranged themselves with the polar heads facing towards. It always formed droplets, oil in water, and the surface tension was much higher than that of cells, called as sandwich model. The model, which was invented in 1935 by Danielli and DAVSON, is also called as sandwich model. In your examination, these models are asked for 4 marks, any model. So give that explanation, brief explanation, and the diagram which is here. Danielli and DAVSON model. This is the lipid bilayer. These all are phospholipids. This is water loving portion. This is water hating portion. And see, in between the plasma membrane, the polar pore is present. This pore helps in transportation according to Danielli and DAVSON model. This is the protein coat which is present on the outermost surface, and this is the lipid bilayer. This type of structure was given in 1935 by Danielli and DAVSON, and it became the first type of plasma membrane model. Let's see the second one. Robertson model. In 1965, Robertson noted the structure of membranes seen in the electron micrographs. He saw no spaces for pores in the electron micrographs. He hypothesized that the railroad track appearance came from the binding of osmium tetroxide or to proteins and polar groups of lipids. Proposed unit membrane hypothesis. See, in the Danielli and DAVSON model, we shown that the diagram shows the pore gate channel type structure from which the transportation takes place. But in 1965, Robertson explained that he saw no spaces for pores in the electron micrograph. So let's see the structure of Robertson model. See here, there is no pore region shown by the Robertson model. Extracellular side, glycoprotein coat is there. Then this is the Robertson model. This is lipid bilayer. Again, this is the glycoprotein coat from the inner side also, but no pore region is present for transportation. So this is the difference between the Daniell and DAVSON model and Robertson model. Now let's see the third one, fluid mosaic model. This is the most important, and recently we are studying still now, that is the fluid mosaic model. So according to S.J. Singer and Gart Nicholson, Singer and Nicholson in 1972, the biological membranes can be considered as a two-dimensional liquid where all lipid and protein molecules diffuse more or less freely. So this is the recently invented, and till now no other research is there for the fluid mosaic model. Singer studied phospholipid bilayers and found that they can form a flattened surface on water with no requirement for a protein coat. That means the outermost protein coat, which was shown by Daniell and DAVSON and Robertson, both the scientists, both the model shows the protein coat on the outermost layer of polar heads of phospholipids. But here it denies that no protein coat is present. It occurs in form of globular proteins, and it is widely accepted model. Till now, fluid mosaic model is widely accepted. It was invented by Singer and Nicholson in 1972. Let's see the structure here. This is the fluid mosaic model. The bilayer, it is shown here. See the upper layer and the lower layer. These upper globular proteins. So see one by one the labeling: carbohydrate groups of glycoproteins. As I have told you previously also, that glycoproteins are attached on the outermost surface of the plasma membrane. Peripheral proteins are loosely attached to integral proteins. Then membrane spanning integral proteins are tightly bound to the membrane. This is the cholesterol molecule, yellow colored, which are present in between the phospholipid molecules. Intracellular surface of membrane. Then membrane splits into layers in freeze fracture electron microscope. So this is all about the fluid mosaic model, which is invented by Singer and Nicholson in 1972. Now I have explained you today here the three different models of plasma membrane, and different diagrams I have also given. So please find the points which differentiate the models and study this lecture once and twice, so that you can understand the functions of plasma membrane, the different models of plasma membrane, who invented those different models, and in which year. So friends, today we will stop here. Thank you very much.